Yeah, I'm really excited to be at the Allen Institute. This is obviously a really happening place in neuroscience. I'm very happy to collaborate with many of you. And I want to say about Clay, that Clay made the quip about being 19, but the thing that's most fun about collaborating with Clay is that he's permanently 5 years old. That, I work, I'm working, I'm tired, I hate making the lab keep on going, and I come and I talk to Clay, and Clay has just got this childlike passion for science, and he gets delighted, and he's got that twinkle in his eye, and so that's what's the greatest thing about collaborating with Clay. In addition to his being smart, but everybody already knows that. All right, so I want to talk about the connectome, obviously. And we are reaching a transition time in the field where a lot of the technological problems that we promised to solve are getting solved. I can't say they're solved yet, but they are getting solved, and we have to think seriously about science. We have the luxury to think about science again, and the question is, what science can we get out of this? Now, my kind of pet motivation for going into connectomics was to investigate learning. And I wrote about this in my book and in position papers a long time ago. These were sort of speculations about the future, but the future is now. And skeptics have always said, you can't use the connectome to investigate learning, you numbskull, because you would need the connectome at 2 time points. Right? You have to have before and after. But I want to argue that that's not the case, that in fact, the connectome at 1 time point might be sufficient to constrain our understanding of how the cortex learns. And the crucial addition is that we need not only the connectome at 1 time point, we need observations of neural activity. That if we combine observations of neural activity and the wiring diagram, then we might be able to constrain the theories of cortical learning. And in particular, every theory in neural nets, in artificial neural nets, theoretical neuroscience, all these theories have predictions about the stationary state to which learning converges to. So I'm not talking about learning in the sense of one episodic memory being stored, but if you look at sensory cortex, you would imagine that maybe it's learning, maybe it's development, but sensory cortex, after being exposed to stimuli for a long period of time, will converge to some kind of stationary state at which it's not really learning anything new. And that stationary state should have some properties which depend on the learning algorithm that got there. And so I'm going to loosely use the term learning. Neuroscientists like to distinguish between development and adult learning. In artificial neural nets, people don't really distinguish between those two. I'm going to assume, as widely assumed, that some of the same mechanisms underlie both experience-dependent development and learning. All right, now, that's the overall big picture of where I want to go in the talk, but let's start with a particular kind of empirical observation, which is, or maybe a principle, which is guiding some kind of empirical observations, and that's the idea of like-to-like connectivity, which is a way of relating structure and function. So no commitment to mechanism here. If you observe the brain, is it true that neurons with similar functional properties tend to be connected with each other with greater probability or greater strength? Right? So if I just look at the activity patterns of neurons, I can see whether they have similar visual responses, for example. And then if I have the connectome, I can see whether or not they're connected. I could look at the number of connections between them, the size of the connections, which is a proxy for strength, and so on. And so for like-to-like connectivity, we would, this is just a cartoon, but imagine that strongly coupled pairs of neurons tend to respond to the same stimuli, whereas weakly coupled neurons tend to respond to dissimilar stimuli. If this is the case, then we would say there's a like-to-like connectivity rule. All right, and there's been a number, there's actually a long history of investigating this hypothesis. And for now, let's put any notion of learning aside, because we can just investigate this empirically, whether or not we believe it's a product of learning. I think the modern era of this kind of study starts with the work of Merschers-Flogel, in which calcium imaging in mouse visual cortex was followed by measures of connectivity through synaptic physiology in vitro. And in a series of papers, this lab showed that pairs of excitatory neurons obey a like-to-like connectivity rule. And then work from Clay's lab came out around the same time as some of those later papers, and they used calcium imaging followed by serial section EM to find results consistent with this idea of like-to-like connectivity. And the rule appears to hold for both the probability of connection and for the strength of connection. Okay, so the next question you might ask is, well, what about inhibitory neurons? That was for pairs of pyramidal neurons in cortex. And inhibitory neurons are just technically harder to study because they're obviously rarer, so that's one big reason. And there's some papers from Rafa Yusti's lab which provide evidence that inhibitory cells synapse in an indiscriminate way on nearby excitatory cells. So if you look at I to E connections, it appears that there's no kind of selectivity, and that seems like it might be incompatible with the like-to-like connectivity rule, which is a kind of specificity of connections. All right, now, but there's a recent paper from Mercer Flogel's lab. This is on bioRxiv. As far as I know, it's not published yet. And this paper took a mouse line where parvalbumin-positive inhibitory neurons were labeled. And they did this kind of experiment of like-to-like. And so you can see right here, each of these points is a pair of a PV positive and an excitatory cell. And the x-axis is the response similarity, so some measure of the similarity of the activity patterns. And then the vertical axis is the strength of the connection. And in this graph, it's the I to E connection, the inhibitory connections. And in this graph, it's the E to I connections. And so you can see right here that, at least for the I to E connections, there seems to be a pretty good like-to-like rule here. So more similar neurons the more similar the neurons' responses are, the stronger the inhibitory connection between them appears to be. And there's an effect here, but it's noisier. So it's not clear whether this is a real effect, but this seems fairly convincing. And so you might ask, well, with our now let's get to the project that we're doing for IARPA, the Microns project, the collaboration by which I've gotten involved with all of these wonderful collaborators in the front row here. And we have neural activity as it was visual responses that were imaged at Baylor by Andreas Tolias's group. And then we have the EM that was done here, and we can try to put together both of these data sets and investigate like-to-like. One small problem is we don't have the activity of the inhibitory neurons. So if you want to look at I and E pairs, you're not going to have any measure of their functional similarity. So we didn't investigate the like-to-like rule as I explained it before. We investigated a different version, and this is like-to-like for common input. So suppose I've got two excitatory neurons, and they're both receiving input from a common inhibitory neuron. And the like-to-like rule would be that if they're receiving strong common input, they'll have similar tuning curves. And if they're receiving weak common input, that's going to be dissimilar tuning curves. All right, so this is a like-to-like rule for 3 neurons, a triple of neurons rather than a pair of neurons. It's clearly conceptually related, but it's not exactly the same kind of like-to-like rule. And so one reason we're doing that is just technical, because we don't have the activity of this neuron. And the other reason is that it's another kind of, you'll see later on, it's another way that we can probe, empirically test theories of learning. Right? The theories of learning can make predictions about like-to-like for pairs, and they can also make predictions about like-to-like for triples. So it's another kind of, it makes our measurements of these like-to-like rules richer if we investigate this too. All right, and it's going to turn out that the specificity, it could be that these like-to-like rules, everyone here knows that inhibitory neurons come in many different types, And so we have to be more sophisticated about, we can't just say inhibitory neurons, we've got to say which kind of inhibitory neuron. And right now, we've been focusing on basket cells because we can identify them by their propensity to make synapses on the soma and close to the soma of pyramidal neurons. So just by looking at where the inhibitory synapses are, we can figure out whether an axon is from a basket cell or not. All right, so you guys have seen, I'm sure everyone here has seen the reconstructions, so I'm not going to really show a lot of that here, but let me just show you some examples of pyramidal pairs getting common input just to make this concrete. So that's a basket cell axon. And you can see that this volume is sort of small, so we don't see the whole basket cell. We don't see the soma or anything. But just because it's making a lot of synapses onto somas, we can identify it as a basket cell axon. And in this case, these two pyramidal neurons are receiving not too many synapses from the basket cell axon. And then we have other examples where the basket cell axon makes several synapses onto both of the pyramidal neurons. All right, now I can show you just anecdotally that because we also have the visual responses of these neurons, we can look at the tuning curves, directional tuning curves. And in this case, this pair of excitatory neurons receiving common input have very dissimilar tuning curves. And in this case, when they're receiving strong common input, they have similar looking tuning curves. Now, this is just anecdotal, right? I'm just showing you two examples. But now I'm going to show you that this is a consistent pattern for a particular class of basket cell axon. And here we have to rely on the ability to classify. So I should say this analysis was done by Sven Dorkenwald in my lab in close collaboration with Nuno and Agnes at the Allen Institute. And a crucial realization that they made was that it's possible to separate these basket cell axons into different morphological classes. And they were, I guess it was before lunch when they were named. But roughly, I don't know, is that the origin story? You know, is before lunch the origin story? So I should ask Agnes. So the Basmati axons, well, they're basically named after the shapes, the typical shapes of the presynaptic boutons. All right, and I'm not going to do justice to this, partially because this classification is still it's being intensively studied by the collaboration. So two kinds of analyses are done. One is sort of by eye. Can a human distinguish between these axons? And then Sven is also trying to automate the classification of the axons by looking at morphological features and so on and so forth. And so the interplay between those two approaches, we hope is going to lead to a classification, but the definitive classification. But I should just say that right now it's still preliminary, but the nice thing is that with many different refinements and changes to the classification scheme, the results typically agree. All right? All right, and here's the analysis that we're going to use to investigate the like-to-like for common input, which is, suppose we've got this axon. Let's take all of the postsynaptic pyramidal neurons, and then let's estimate the strength of the connection for each postsynaptic partner. And we'll do that by quantifying the area of the synapse, basically the PSD size. And then if there's multiple synapses, we add up all those areas, and that gives us an estimate of the strength of this connection, morphological estimate of the strength of a synaptic connection. So for every one of these postsynaptic partners, we have a connection strength, and then we threshold that connection strength. We divide the pyramidal neurons into a group that are strongly connected with this axon and a group that are weakly connected with this axon. And then, for every pair of neurons in this strong group, we compute the Pearson correlation coefficient between their tuning curves. That's a measure of response similarity. And that's shown here. I have 3 Pearson correlation coefficients, and then we can just average those Pearson coefficients together to get an overall measure of similarity within that group. And there's also selection criteria, so just for you to realize what we do here, we have to get the cleanest results, what we do is we retain the pyramidal cells that have more reproducible visual responses. So there's various ways of doing that. For example, a coarse way is just by choosing the neurons that show a very tuned tuning curve. But we're pursuing other means, for example, looking at simply reproducibility, looking at the first half of the experiment and the second half of the experiment to see if the tuning curve is the same. And so that winnows down our cells, and those are the cells that go into this analysis. Oh, sorry, I should have mentioned the stimuli. So they are oriented stimuli. They're not exactly gratings, but they're, I forgot what Baylor calls them. Monets. So they're sort of filtered noise, which turns out to be oriented. Yep.
Do you literally kind of include the cases that all the trillion neurons, they don't have the connectivity between them, or you don't have any constraint? You just don't, constrain whether or not.
You mean, do we throw out the neurons that are not connected? For any given axon, we only consider the neurons for which we can see a connection. All right, some of the neurons could be connected outside the volume, but we don't.
Put any constraint as long as they share the common kind of like.
Well, first we constrain this. So these cells all have functional data, and it's sort of repeatable. So we winnow that down. Then for every one of these axons, we take the subset that are connected. And this is now showing you 3 pancake axons. So each of these columns is a pancake axon. And then this row shows you the tuning curves of the weakly connected postsynaptic partners. And this row shows you the tuning curves of the strongly connected postsynaptic partners. And the black right here is showing you the average of the tuning curves. And one thing you can see here is that the preferred direction in this particular volume is somewhat biased, which is a known phenomena. It's known that there can be a bias toward the cardinal directions. This is a fairly strong bias. It's just perhaps the luck of the draw in where this volume was chosen. But in all 3 of these cases, if you look at the lower row, the strongly connected postsynaptic partners obviously have more similar tuning curves to each other than the weakly connected postsynaptic partners. So on an axon by axon basis, we can do this. If you look at a bunch of pyramidal cell, you look at a bunch of basket cell axons of the pancake type, this is the average similarity of the weakly connected postsynaptic partners, and this is the average similarity of the strongly connected postsynaptic partners. And if you just look at the pairing, it's very convincing that for every axon we see this effect occurring. Now, on the other hand, if we look at the Basmati axons, you don't see any consistent trend. And if you look at the potato axons, you don't see a consistent trend either. So I'm cautioning you, these are still preliminary results. We're still trying to get them as solid as we can. But I found this kind of paired comparison was what it ultimately convinced me that we're going in the right direction. And I find these results believable. OK, and so I'm going to switch now. So all of this was really about the first kind of like-to-like results that are coming out of this analysis of this reconstruction of the phase 1 microns project. And I've already thanked collaborators at the Allen Institute at Baylor. And I want to emphasize also there were a lot of people involved, not just in, Sven did the science in collaboration with Tommy and Nick. But there's plenty of other people who worked on the actual computer science of the reconstruction. And unfortunately, I'm not going to have time to recognize their efforts by describing all the things they did to get where we got. I'm going to go out on a limb and tell you about a little theory that might make some sense of what this like-to-like means. So, let's say that this like-to-like result holds. Let's say it's really true in the cortex for some kinds of inhibitory neurons, for pyramidal neurons, and so on. Is that surprising? What do theorists say about like-to-like? Right? That's the first, in the history of theoretical neuroscience, is it somehow obvious that we should get like-to-like from the theories? And the first thing I'd like to say is that in the classic models of cortical development, it falls out. All right? And so let's go back to 1973. This is the first model of development of the cortex by Christoph von der Malsburg. And what he assumed was that there's some kind of sensory afferents from the thalamus to the cortex, which are Hebbian. Neurons that fire together, wire together. And then he also assumed that there's some intracortical connections, cortical-cortical connections. And they're fixed. They're not modifiable. But they have a spatial organization. So the excitatory E to E connections are strongest when neurons are right on top of each other. And the strength falls off as you move farther away in the cortical sheet. So remember that the cortex is like a sheet. And so we can, if we have two neurons in that sheet, we can measure the separation between those two neurons. And so the idea was that the strength of excitation falls off with separation. And also inhibition is the same way. Strongest inhibition when neurons are close together, and a weaker inhibition when the neurons are farther apart. And this decay is, this space constant is longer for inhibition than it is for excitation. And von der Malsburg did this to explain the existence of feature maps, orientation maps in visual cortex. So this is a famous picture from Gary Blaisdell's lab showing, the existence of orientation maps was already known, but this was, I think, the first time it was shown by imaging. I'm not sure if that's the history. Is that the right? Hopefully that's the right history. But you can see right here that every location in the visual cortex of this monkey or cat, I can't remember, cat, is encoded by its preferred orientation. And so nearby cells have similar preferred orientations, and there's some kind of slow changes as you move inside the cortical sheet. And if von der Malsburg already showed in 1973 that you get this kind of feature map if you simulate his model with fixed intracortical connections, spatially organized intracortical connections, and Hebbian sensory afferents. All right, so that's the basic outlines of the model, and it's been repeated and refined for a long time by people like Kohonen and Ken Miller, and so on and so forth. Totally classic kind of model. And so here's the trivial consequence of that. So an orientation map or a feature map means that feature space is aligned with physical space. Right? Nearby in feature space and nearby in two-dimensional cortical sheet are the same thing. And in this model, connectivity is spatially organized. So nearby neurons are connected. So it follows that neurons that are nearby in feature space are also more strongly connected. Trivial consequence of the assumptions of the model. All right, so the von der Malsburg model leads to like-to-like connectivity for both excitation and inhibition. Now, what's the problem with this? Well, it's great, but mouse visual cortex is generally thought to have a so-called salt and pepper organization. So if you look at mouse visual cortex, and I think this was a paper from Clay's lab that showed this with imaging, you don't see this map what you see is an apparently random organization, that the preferred orientations of neurons are random inside 2D space. So it doesn't seem like these models can be applied, at least naively, you can't apply them to explain mouse visual cortex. And not only that, even if you want to explain cats or primates or humans, there may be areas of cortex that lack feature maps, right? Primary visual cortex is only one cortical area, and it's not clear whether this principle, this kind of model, generalizes to anything that doesn't have a feature map. So that motivates exploring some other class of models. And so what I want to do here is investigate models where the inhibitory connections are plastic and driven by so-called anti-Hebbian plasticity. And the model I'm going to talk about is most similar to, the most similar model in the literature is a spiking model due to Mike DeWeese's lab, which separates excitatory and inhibitory populations and endows them with these kinds of plasticity rules. But there's a long history of rate models. I'm going to use a rate model, a non-spiking model. And so in that respect, it's like this long history of models, most recently explored by Mitya Shklovsky and collaborators. But these other models haven't made a true split between excitatory and inhibitory populations. So that's what we're going to add here. And we're going to compare it with biology. But the comparison with biology is going to be very qualitative. So it's not worth doing something really quantitative in detail right now because the data is still mushy, and we're still looking for principles, qualitative principles. So here's the architecture. So suppose we have these sensory afferents. We've got excitatory neurons, inhibitory neurons. Every green arrow here is an excitatory connection. Every red arrow is an inhibitory connection. The green arrows all change by a Hebbian plasticity. So neurons that fire together wire together. The red arrows also change in the same way. So correlated activity of these two neurons will cause this inhibitory connection to strengthen. But following Peter Foldiak, we call that anti-Hebbian inhibition. And the reason is that inhibition is like a negative synapse. So you're strengthening an inhibitory connection is like making a negative number more negative. So Hebbian plasticity means correlated activity drives the synaptic strength in a positive direction, whereas anti-Hebbian means that it drives the synaptic strength in a negative direction. But in both cases, it's neurons that fire together, wire together. And so the difference now, okay, we're neglecting important classes of connections that are known to exist. We're neglecting E to E connections. We're neglecting I to I connections. We're neglecting the diversity of inhibitory neurons. And the reason is we've thrown out everything that's peripheral to the question we want to answer here, which is this interaction between excitatory and inhibitory populations. How could anti-Hebbian plasticity function in that interaction? All right, so I'm going to show you some simulations, and this is a rate-based model. Let's see, I'll show you. I guess I can show you the equations. So this is the set of equations. X are the excitatory activities, Y are the inhibitory activities. A is a matrix of the connections between excitatory and inhibitory populations. W is the matrix of connections from the sensory afferents to the excitatory neurons. And here are the plasticity rules. So we've got this excitatory connection, which has a Hebbian thing. And E to I also has this anti-Hebbian term, which is just correlated, driven by correlated activity. And there's a homeostatic term. So there's a synaptic scaling that occurs when it functions to keep the activity of the neuron at some kind of set point. And then these terms are synaptic competition. So there's a lot of evidence that in neuroscience that synapses that converge onto the same neuron or diverge from the same neuron may compete with each other. So that if one synapse goes up, other synapses have to go down. And so that's this competitive term inside the synaptic plasticity rule. And it turns out these competitive terms are very important. So Heb by itself is not enough. There's a long tradition of adding these kinds of terms that are not activity dependent in order to satisfy other properties that are necessary for a learning model. And we have non-negativity constraints on these synapses to make sure that they really are excitatory or inhibitory. All right, and I'm going to go in the style of 1990s machine learning and use the MNIST data set of handwritten digits here. And I'll have 64 neurons here and 5 neurons here. So here's the classic MNIST data set, familiar to all machine learning researchers. What we do is we run the network dynamics until it gets to a steady state, and then we update the synapses. And we do that for every one of these visual stimuli. OK, so here are the, after the network has learned for a while, here are the features that are learned by the excitatory neurons. So these are the feed-forward connections of each one of the connections from the sensory neurons to the excitatory neurons. And they have the same dimensionality as the images, so I can look at them as images. So you can think about these as the receptive fields of those excitatory neurons. So the network has clearly learned something. It's learned things that look like strokes. It's been shown handwritten digits, but it's learned these kind of stroke-like features. This is sort of old hat from 1990s unsupervised learning, so I'm not trying to impress you with this, but just to show you that it's learning something. And this graph now shows that it looks at the correlations between the E cells after learning. And so what you can see right here, this is the Pearson correlation coefficient, and this is the number of excitatory pairs that have that Pearson coefficient. Most of them have a Pearson correlation that's close to 0, but you can see that there's some tail of neurons that have more strong correlations. So the correlation between pairs of neurons can vary. And this is the result of doing the like-to-like for common input analysis. So I've got 5 inhibitory neurons in my model, and I take the 64 excitatory neurons, and for each of those inhibitory neurons, I take the 5 most strongly connected excitatory neurons, and I say that's the strong group. And in each case, you can see that the response similarities for the strong group are higher than the average response similarities for the weakly connected group. Now, numerically, it's not the same thing, but qualitatively, you can see that this like-to-like for common input holds for this model. Yeah, question?
No question. When you actually show this sparse structure, is that mostly because of the actual algorithm or because of the non-negative constraint algorithm?
Well, the non-negativity is part of the algorithm. So the non-negativity and the synaptic competition.
But if you relax the weight matrix, be able to take any number, then do you still see this kind of like
No. Well, OK, I should say the following, that I'm not going to get into this, but if I change parameters, I can make this. So if I change this ratio, which this is, P and Q controls the amount of synaptic competition for the inhibitory synapses, I can make these features look more like strokes or more like whole digits. So the sparsity is controlled by that synaptic competition rule. But it depends on having the synaptic competition and the non-negativity. And I had a long-term interest in saying that non-negativity of connections is a computational thing. Right? That's the whole point of non-negative matrix factorization and so on. All right. So that's showing you that we can reproduce this like-to-like for common input. And here is like-to-like for the direct connections. So I take all the EI pairs in the network, and I look at the correlation coefficient between their activities. And that's on the vertical axis. And I look at the connection strength between the inhibitory and excitatory neurons, and that's on the horizontal axis. And we can see that there's this relationship between correlation and connection. So we have like-to-like for direct connections. This is similar to what this preprint from the Mercer-Flogel lab. And here's just showing you, just to give you an idea of the activity pattern. So this is 64 excitatory neurons, 5 inhibitory neurons. And these are 100 stimuli. And you can see that the activity patterns of the excitatory neurons are quite sparse. And so these are more selective.
And the inhibitory neurons are less selective. They're active all the time. Here's showing you a kind of bonus, which might not have been obvious to begin with, but this is imagine I were doing voltage clamp on my excitatory neurons, and I could separate out the excitatory input and the inhibitory input for each neuron. So I've got 64 excitatory neurons. In green is the excitatory input, and in red is the inhibitory input. So the active neurons are only those for which the green exceeds the red. And basically, it's the difference between the green and the red, which is the activity of that excitatory neuron. And what you can notice here is that when green exceeds red, it usually exceeds it by a small amount. So E and I are roughly the same. And so here's a graph. I just take the same data here, and I graphed it like this. Here's excitatory input versus inhibitory input. And here's the graph where E is exactly equal to I. This triangle below is where the neurons are active. And you can see that these points are all close to that diagonal. So we have approximate EI balance for active neurons, which is something similar to what's been observed in mouse cortex.
This
Yeah.
is the effective input for a particular stimulus, or just the anatomy?
No, this is for a particular stimulus, looking at all the excitatory and inhibitory input. Okay, so I can do the stimulus, but I can present a different stimulus. This graph will look different, and these points will look different, but you'll see the same pattern. All right, so I showed you a theory, and I don't have time to really get into. I just showed you the highlights of a model that reproduces like-to-like for both excitatory and inhibitory. And your reaction might be, oh, okay, well maybe it's easy to get like-to-like. So what I want to show you now is another theory of the cortex, which is not consistent with the facts. And that other theory is one which has become very trendy now. And that's the idea that the cortex performs backpropagation. That the algorithm which has been driving the revolution in artificial intelligence is precisely the one that's used by the cortex. And in fact, I wrote a paper like that with my student Xiaoyi in 2003 where we explored that. We found some interesting, we argued that we thought we had a biologically plausible implementation of backprop. I actually gave up on that idea. And at the time, I kind of despaired about figuring out what learning algorithm the cortex uses. And that's why I started working on connectomics. Right? In the meantime, in fact, I love backprop. I've been using backprop since 2006 for the Connectome, even when everyone said it was stupid. Right? But in the 2000s, all the theoretical neuroscientists said backprop is stupid. And now they say, oh, it's great. That's the way the brain works. So that's the reversal that's happened. And to be fair, there's a really interesting paper by Lillicrap. It's out of DeepMind called Feedback Alignment, which is trying to make backprop more biologically plausible. But why don't we use the Connectome to test this idea? And I want to say that these papers, they basically, their concept is that the cortex, although it has recurrent connections, those recurrent connections are just there to assist the training of a feed-forward net. So we've got a multi-layer perceptron, and there's all these other recurrent connections, but they're just there for the learning. They don't really do anything for the computation. And the reason that perspective is taken is that if you want to train a recurrent net with backprop, the backward pass actually goes backwards in time. And so that seems really biologically implausible, if not physically implausible. So that's why we're going to just consider this possibility that we've got a multi-layer perceptron trained by backprop. All right, so here's the experiments. Again, I'll just do I'll do an experiment in the flavor of a homework assignment in my deep learning class. I'll just take a multi-layer perceptron, which takes the MNIST handwritten digits as input, and it's got 2 hidden layers of 512 neurons, and it's got 10 output neurons, which just classify the digit class 0 through 9. I'm going to make this network a little more biologically plausible by implementing Dale's law. So I flip a coin with 80%, 20% probability, and I decide ahead of time whether a neuron is excitatory or inhibitory. So in every layer of the net, we've got 80% excitatory neurons and 20% inhibitory neurons. and we constrain the outgoing synapses of those neurons to obey Dale's law. Turns out that that doesn't matter for the results I'm going to show you, but it's something that people always ask you. And we train it, and then I'm going to show you graphs from the second layer of weights. So we're going to compare the activity, pre- and postsynaptic activity in this layer, and we're going to compare it with the connections between these two layers. So here's the first graphs I'll show you from this experiment, which look at the postsynaptic partners of an excitatory neuron. So suppose I take an excitatory neuron, and I look at all of its postsynaptic partners, and we graph response similarity versus connection strength. And you can see that this is like to like, right? More similar responses correspond to stronger connections. And that was for one excitatory neuron, but I can compute the slope, the sort of best fit line for. I could do this graph for every excitatory neuron in my network, and then in that second layer of my network, and I can fit a line here, and that's this correlation coefficient. And so you can see that they all have positive slope. It's consistent with this like to like. So it's not just this one excitatory neuron and its partners, but the rule holds for in general.
But this is feed forward like to like. It's not.
That's right. It's feed forward, because that's the model that people are.
Diesel rather than research flow loss.
Well, that's right. And it's different. It's not a straightforward comparison with the model I showed you before, which has recurrent connections, recurrent dynamics. All right, but this is as I remind you, that's the theory. The recurrent connections in the cortex are there to somehow enable backprop training of a feed-forward net. That's the kind of theory that's being proposed these days by theoretical neuroscientists. And so the first thing I'll have you notice is that like to like, you might think that like to like for excitatory pairs, that's got to prove that Hebbian learning is working. But there's no Hebbian here. It's backprop. So it's actually not constraining. Right? Like to like between excitatory neurons is not very constraining. Both theories satisfy qualitatively. So let's now look at inhibitory neurons. So I look at the postsynaptic partners of an inhibitory neuron, and what do you see here? Well, stronger connection means dissimilar activity. So unlike neurons are connected. All right, so this is inconsistent with the like-to-like finding from functional connectomics. And it's different from the like-to-like conductivity that we see in the anti-Hebbian model. So here's a theory in which like-to-like does not hold, at least for inhibitory connections. And so this is something that it didn't really seem obvious. I didn't really get it. But like-to-like for inhibitory connections is very different than like-to-like for excitatory connections. I thought, oh yeah, it sounds the same, right? But they're really different. And these models show that. Yeah, question?
Yes, on one side, you haven't commented on the previous graph. You have, there is no like-to-like for common input, but there is like-to-like for the feed-forward part.
That's right. So in both cases, whether it's excitatory or inhibitory, if we do the common input analysis, no like-to-like rule.
But aren't these, so is that part of statistical power for being able to tell this? Because these should go hand in hand together, right? You've got like-to-like, assuming that you push it to the limit of very large number of neurons, wouldn't automatically like-to-like for the feed-forward generate a like-to-like for the common input?
OK, so the first caveat is that I did these simulations a few days ago, so maybe that's wrong. I think it's right. And so what's interesting is that this shows you now, remember I argued that like-to-like for direct connections and like-to-like for pairs and for triples, these are somewhat independent tests of the theory. The triples add a richness. And so what this is showing to you is that you can have like-to-like for direct and not have it for the common. I mean, it's a little, if you think about the theorems about correlations, it's not impossible.
It's not impossible.
It's not, correlations are not transitive.
Yes, but it would require, so if you only have the second order correlation, you'd expect to find a third order correlation, but you can probably build a network in such a way where you can actually have a cumulant property for these where you take the third order away from it. But at least on to naive thinking, you'd imagine that the left side would imply the right one.
Naively, you would.
No, that's the third order. The right is the third order.
That's right. That's right. This is the second order. This is the third order.
Yes. So naively, you'd imagine that the second order would imply the third order.
Naively, but no, because this is not. So that's the thing, is that a Hebbian theory is really correlation based. You're going to find all kinds of higher order correlations in Hebbian, but backprop is not really correlation based learning rule. So it was kind of interesting that you can't distinguish Hebbian from backprop based on the second order, but probably based on the third order you can. So that's showing you, I think, the power of adding a third order version of the rule. And so here also we don't get it for inhibitory connections. All right, so this is then, obviously this is one particular instantiation of the backprop idea. If I tell a backprop enthusiast about this, they'll say, oh, I'll modify the theory then, right? And I'll make the inhibitory neurons governed by some other rule. But that's kind of the point, that at least we can knock down some theory of learning, right? So we have some possibility of falsifiability, which we didn't have before functional connectomics, right? So I think that's an important, before the question of whether the brain uses backprop is kind of subjective. It's just different theorists have different intuitions about what's biologically plausible, right? But I think now we have the potential for testing theories empirically, and that was the reason. That's really, if you go back to over a dozen years ago, that's the reason. That's what drew me into this. And I didn't think it would take 12 years to get to the point where we could test these theories. And then this is just to show you then that I apologize for this plot, but this is again the test of the EI balance. So this is the excitatory versus inhibitory input scatter plot for a layer of neurons. And you can see right here that these are the active neurons, and they're not close to this diagonal. So EI, there's no reason, if you think about it, EI balance, there's no reason it should hold in a backprop trained multi-layer perceptron. And in fact, it doesn't. All right, and so I'm going to just skip over the various, there's interesting optimization principles. You can get that network model from various kinds of formal optimization principles for unsupervised learning. I'm going to skip over that. But I want to argue that in this kind of broad strokes, even though we haven't added the other classes of connections yet, we don't have the diversity of inner neurons, we get these biological facts from the Heb, anti-Heb, the first model I talked about. So we have sparsely active excitatory neurons. We have, this is a misprint here. This is the sensory to excitatory connections. They're sparse. We have densely active inhibitory neurons. We have E and I connections are dense and antisymmetric. And we have like connects with like. So I feel like this is an interesting line of theoretical research. And as connectomics progresses, I think we will be able to refine our tests of models of cortical learning and development. So I guess I'll wrap up there and take questions. Thanks very much. Okay.
I have a question regarding the common input, the inhibitory common input. I noticed that it has also kind of related to convolution here, which is different from what you were talking about versus global data.
Mm-hmm.
In the triple neuron case, like only applies to the two excitatory neurons. The inhibitory neuron doesn't have, in your model, it doesn't have to have the same tuning properties of the excitatory. As long as it has the common strong input for the excitatory, it could work. Is that right? Or no? Or you have to have the inhibitory
Okay, yeah.
neurons also have the similar properties.
And Sebastian, could you briefly restate the question for the recording before you answer?
Let's go all the way back. All right, so this,
The like for like, yeah.
so this really is a question of, so like to like is just a kind of regularity that we observe in the data. Now the question is, what's causal? So in activity-dependent plasticity, activity causes connections, right? That's the learning explanation. But there's another explanation, which is that connections cause activity. And so what you're saying here is, well, obviously, if this inhibitory neuron is providing really strong drive to both of these neurons, then they should end up being correlated. Their activities should end up being correlated with each other, as opposed to the other explanation.
I don't think that
No, that's not your question.
You're saying it's like to like. She's saying it's X to like. We don't know what X is, and you're assuming
No, I said this is, it's kind of a
it's
logical
something to
tool.
like.
No, wait, was that your question?
My question is, does the inhibitory neuron left need to have similar activities as the pair of excitatory or not? The like for like, the two excitatory are like each other.
Right.
Similar tuning curves or
Right.
whatever. But the inhibitory neuron doesn't have to have a similar property to the other two as long as it has strong, it just somehow synchronized the
Well, this particular statement of the principle doesn't commit to whether this has similar tuning properties. It doesn't commit to it. Now you're asking whether in reality that's true? Yeah. Well, yeah.
No, it's an excellent point.
Well, okay, but if you believe the Mercer-Flogel result, then if you combine those two, right? Yeah. Great question. Yeah.
For your model, the results you found can be results from the statistics of your inner stimulus, or these static plasticity constraints with focus spinning or snaps competition. So I think if you feed your model with noise or get some spontaneous activity, let it converge, can you still see the same like to like rules in the model, or it's heavily dependent on your inner stimulus?
It's, well, I don't know. So the direct version of like to like you will see. And the reason is that it's sort of, it's implicit in the Hebbian assumption. Right, so at a stationary state of learning, delta W on average is 0. And so then you'll see that this has to roughly equal this. The expectation value of this correlation has to equal WIA. So it's not surprising in the sense that it's kind of put in by hand. Once you assume that it's Hebbian, once you assume that learning is correlation-based, then connection strengths have to end up looking like correlations. Yeah, but I can't make that claim about the common input version. I don't know about that version. I don't have any. I have an analytical theory for the direct connection version. I don't know how to do it analytically for the triple neuron version.
But at least you can try to speed the white noise.
That's right. Yeah. Stefan?
For the EM analysis, you compared the tuning for weakly connected and strongly connected. But in a sparse connectivity matrix, the weakly connected ones would actually be the nearby ones where there are no connections. Would you consider doing a similar analysis based on touches with no synapses as being the null hypothesis rather than the weak connections being the things to compare?
Right. So what we examined was if we take the connected neurons and we just let's see which ones are more strongly and which ones are more weakly connected. Now the other way to do it would be just compare connected neurons versus not connected neurons.
But neurons which can potentially be connected.
Yeah, nearby neurons.
Constraints.
Right. Well, we haven't pursued that. So, Mercer-Flogel, their lab reports that Rafa used to is right, that basically everything is connected. So that's sort of their, yeah, we've done proximity controls. Yeah, so we can do, I didn't show that, but we have the proximity control we've done is to have look at pairs where one of these is a near miss, right? And what you see then is that the similarity is lower. But we haven't really pushed that. So I don't have a I'm not ready to make a claim about that connectivity versus no connectivity. All right, but I do want to say that pancakes are the class of, so if you look at the baskets, pancakes, you look at the pancake basmati potato, the pancakes are the class where you see this morphological estimate of connection strength has the biggest dynamic range. Yeah, question?
So for the like-to-like rules reported in the literature, they seem to fall in a certain part of the spectrum that in principle would occur. So on the one end of the spectrum, it's flat connection probability depending on the similarity of responses, so no like-to-like, right? And on the other end of the spectrum, it's complete like-to-like when only the neurons with the most similar responses are connecting and everything else is not. But what is observed experimentally is that, let's say, neurons prefer the same orientation, or if they prefer orthogonal orientations, the difference in connection probability is about a half, right? It's a factor of 2. So I'm curious, do you have any thoughts about why it's in this sort of middle of the spectrum? You know, if you imagine that, if you think about reasons why you want neurons that prefer similar stimuli to be stronger connected to each other, but why do you have this connection between those that don't prefer similar stimuli, and why to that extent between them?
You're talking about excitatory pairs? Yeah, I don't. That turns out to be in the kind of models I showed, that's controlled by a parameter. So depending on how strong you make the synaptic competition, you're going to end up thresholding out. So basically, every neuron is connected to the neurons that they're most strongly correlated with. But it's a parameter which controls how fierce that competition is. Do you just only talk to the ones that are most similar to you, or are you more less discriminating? So, yeah, I don't. There's a lot of other. I mean, there's a lot of other things. Like if you look at the Mercer-Flogel papers, exactly how they calculate response similarity. Also, so there's a lot of things, details that are lurking below the surface that have to be straightened out.